All right, let's get right into it. Think about your own DNA as a kind of personal instruction manual. Now, what if that manual has a typo? Just one tiny error that could be the key to a mysterious illness you've had your whole life. For the longest time, finding that one typo was like finding a needle in a haystack. But that's changing. Today, AI is becoming this incredible detective, cracking our genetic code to finally diagnose rare diseases. So in this explainer, we're going to look at the amazing promise of this tech and the really big ethical questions it forces all of us to ask.
For anyone with a rare genetic disease, this number isn't just a statistic. It can be a lifetime of not knowing. This is how long it typically takes to get a correct diagnosis. This whole process is so common, it actually has a name, the Diagnostic Odyssey. And it's exactly what it sounds like, a grueling journey of tests, specialists, and wrong turns that can go on for decades.
And this quote from Genome Research, well, it just cuts right to the chase. This isn't just some inconvenience. It's a massive, crushing burden on patients, on their families, and on our entire healthcare system. The human cost is it's immense. And that's exactly why the race is on to find a faster, better way.
And you know what? A breakthrough did arrive. A huge technological revolution that promised to solve the speed problem once and for all. But in doing so, it created a totally new challenge, a massive, overwhelming flood of data.
This technology is called next-generation sequencing, or NGS. Probably the easiest way to think about it is as a super fast, incredibly powerful speed reader for our DNA. All of a sudden, we could affordably read a person's entire genetic code in just a matter of days instead of years. This was a complete game changer.
Now, when it comes to NGS, there are two main approaches. You've got whole genome sequencing, or WGS. That reads everything, the entire book of you, cover to cover. But then there's whole exome sequencing, or WES, which is a lot more targeted. It focuses just on the parts that actually code for proteins, because that's where most of the known disease-causing typos are found. For a patient, this is a really important choice. WES is often the smarter, more cost-effective first step, while GWGS is that deep dive you do when the answer is really hard to find.
But here's the catch. Reading the entire genome, or even just the exome of one person, creates a colossal amount of information. Suddenly, the problem wasn't about getting the data anymore, it was about trying to make any sense of it. We'd basically solved one bottleneck only to create another, even bigger one, a tsunami of genetic data that no single human could possibly analyze on their own.
So, that brings us to the next part of the story. We've got the book, but how do we find that one critical typo? That one single genetic variant that's causing the disease, hidden among millions of other perfectly harmless little changes? That is the central challenge.
Well, that process has a name. It's called variant interpretation. And it's really part science, part pure detective work. It means combing through all that data, checking it against databases, weighing all the evidence to decide if one specific variant is the bad guy. This is, without a doubt, the hardest and most time-consuming step in the entire journey.
So to bring a little order to all this chaos, back in 2015, the American College of Medical Genetics and Genomics, or ACMG, published a set of guidelines. They created this 5-tier system for classifying variants. It goes all the way from pathogenic, which means it definitely causes disease, down to benign, which means it's harmless. This pretty much became the official rulebook for geneticists everywhere.
But here's the thing. Even with this great new rulebook, the process was still painfully manual. It could take an expert hours, sometimes even days, to research and classify just a single variant. This manual work became the new bottleneck, keeping diagnoses slow, making them expensive, and leaving patients stuck waiting.
To break through that bottleneck, we needed a new kind of detective, one that could analyze all the clues at superhuman speed. And this is where artificial intelligence finally enters the story, promising to automate all that painstaking work that humans were doing.
And we're not talking about science fiction here. A whole new generation of AI-powered tools has popped up, with names like Franklin, Varsum, and GeneBee. These platforms are designed to automatically apply those ACMG guidelines, sifting through mountains of data in seconds to find the variants most likely to be causing a disease.
But how well do they actually work? Well, this chart shows you both the promise and the peril. For the really clear-cut cases, the variants that are obviously good or obviously bad, these AI tools are very accurate. But for that tricky middle category, the variants of uncertain significance, or VUS, their performance just plummets. They often disagree with each other, and they disagree with human experts. And that's because for these really rare variants, the science itself is still evolving, which means the AI's training data is often incomplete or just plain ambiguous.
And this quote right here, it spells out the real-world risk. According to a major study, a lot of these tools have a dangerous habit. They tend to incorrectly label these uncertain variants as pathogenic or disease-causing. And that's a huge clinical concern. It could lead doctors to recommend surgeries or treatments that aren't needed, cause incredible anxiety for patients, and rack up healthcare costs, all based on an AI misreading some fuzzy data.
And that very real potential for error just highlights how high the stakes are. The fact that AI can unlock our genetic codes so quickly raises an even bigger set of questions. I mean, your genome is the most personal, most sensitive data that you have. Once we unlock it, who gets to see it? Who's guarding those secrets? This is where the technical challenge becomes a really profound ethical one.
So let's just start with the basics. Where does all this massive amount of data actually live? Is it on a server in a hospital basement somewhere? Is it sitting in the cloud, managed by some giant tech company? The security of our most fundamental information, both physical and digital, is the first giant hurdle.
As you can see here, there are a few different models, and each one has its own trade-offs. You can have on-premise servers, which gives you complete physical control, like a safe in your own house, but they're super expensive and hard to scale up. The cloud is scalable and cheaper, but you're essentially handing control over to a third party. And then there's this new idea of decentralized systems, which could give control back to patients, but the tech is still really new. The bottom line is, there's no single perfect answer.
Okay, so let's say the data is stored securely. The very next challenge is privacy. How can researchers use all this data to find cures and make new discoveries without revealing the identities of the people it belongs to?
People are working on some pretty clever strategies for this, but as you can see, none of them are foolproof. Things like k-anonymity try to hide you in a crowd, while differential privacy adds a kind of statistical noise to mask your identity. The problem is, every method involves a trade-off. The more you protect privacy, the less useful the data might become for the actual research. It's a constant, delicate balancing act.
And all of this leads to a fundamental clash in our values. If you're in the insurance industry, actuarial fairness means you group people by risk to set your prices accurately. It makes business sense. But from a social point of view, is it fair to penalize someone or even deny them coverage because of a genetic risk they were born with and have no control over? This clash between two totally different definitions of fairness is right at the heart of the whole debate over genetic discrimination.
So as we start to wrap this up, we've arrived at the central tension of our genomic future. On one hand, you've got the incredible, amazing promise of personalized medicine and super-fast diagnoses. And on the other hand, you have the peril of misuse, discrimination, and the loss of our most private information.
And this challenge is just getting more and more complex. With what's called multiplex testing, a single blood sample can now be tested for hundreds of different conditions at once. So, how can a patient possibly give truly informed consent for every single one of those tests? The old model is basically breaking down, and it's forcing us to completely rethink how we make sure patients stay in control of their own health decisions.
And that really brings us to the key takeaway. Our ability to read and interpret the human genome has raced way, way ahead of our ethical and legal frameworks to manage it. The tools are powerful, no doubt, but they're just tools. The crucial work now is building the systems of human wisdom and oversight to make sure this incredible power is actually used for good.
So we'll end with this question. The genetic code isn't a total mystery anymore. We have the key, but now we face an even harder task, deciding who gets to write the rules for how this knowledge is used. And it's not just a question for scientists or ethicists. It's a question for all of us as we decide what kind of genomic future we actually want to build.